So it's my great pleasure to introduce the final speaker for today's seminar, Thomas Tullius. Uh, Tommy did his undergrad at Oberlin College before moving to Boston to Harvard Medical School for his PhD with Sterling Churchman, where he did some fantastic work on how RNA polymerases reshape chromatin architecture on individual fibers, which I believe we'll hear about today. And he's since recently moved to Princeton to do his postdoc with Dr. Michael Levine. But with that, I'll pass it over to Tommy to tell us about his work.
Thanks so much for the intro, and thanks to the organizers for inviting me. I've been watching these seminars for a long time, so it's pretty exciting to get a chance to give one. So yeah, I'm going to be talking about some of the work that I did during my PhD in Sterling's lab, which we recently published in Mole Cell. So this is all published work, but if you want to read more about it, you can check it out. I'll have a QR code at the end. And the focus of this work is really on understanding how transcription is influences and is influenced by this really complex chromatin environment in which it occurs. So the focus of this work is really on early steps of gene regulation and of transcription, specifically, the formation of the pre-initiation complex where pol2 is recruited, and then the promoter proximal pause where pol2 is released, and then we'll pause a little bit downstream of the TSS as a sort of additional regulatory checkpoint. Varies from gene to gene how much this occurs. Then from there, pol2 will be released into elongation. And as I was mentioning, transcription, as I'm sure everyone here very much appreciates, is not occurring on some just naked DNA template with nothing on it. It's in this, it occurs on a genome that's packaged into nucleosomes, which can serve as barriers to transcription. It is surrounded by other transcription, both at the same gene, multiple rounds of initiation. You can have other genes, especially in Drosophila, which is a system that I'm working in, very nearby to the transcription that's occurring. You can have tRNAs, you can have enhancers, all of this transcription, all of these nucleosomes. All this is happening at once on these single chromatin fibers. And it's all organized within sort of a linear and 3D arrangement of the genome. But the methods that we have to really probe these kind of single molecule relationships between individual polymerases and this broader chromatin landscape are somewhat limited by the fact that we don't really have good existing genomic approaches tend to be based off of short read sequencing. And so we can use something like MNA-seq to look at the overall distribution of nucleosomes, but we're inherently going to be averaging across millions and millions of cells. And so we're losing the underlying patterning and variability that really tells us a lot about how then, polymerases and nucleosomes and such are directly interacting with each other. So my goal with this work was really to establish an approach to look at this kind of single molecule relationship between transcription and the chromatin landscape using a single molecule genomic approach called FiberSeq in Drosophila S2 cells. So what is FiberSeq? FiberSeq was developed by a former postdoc in Sterling's lab, Andrew Sargakis, who now runs his own lab doing really cool stuff with FiberSeq at University of Washington. And the basic idea here is, like a few of these other recent single molecule genomic approaches like SAMOSA and SMF, is to basically, instead of doing sort of the traditional approach of degrading all the accessible DNA, instead we're going to actually just mark it, in this case with a non-specific DNA adenine methyltransferase, which will add M6A residues throughout accessible DNA. And the nice thing here is that we leave DNA intact, and we have a mark that can be directly detected through PacBio sequencing, meaning that we can read out 10 to 15 kilobase up to like 30 kilobase reads where we've marked accessible bases in a fairly saturated way with M6A residues. And so the net result are these long reads with methylations marking accessibility where you can very clearly see nucleosomes. I was interested in going a step further and really trying to hone in on not just nucleosomes, but also other footprints that are left by the chromatin-bound proteome in this data, like the promoter proximal pause pol2, transcription factors, et cetera. And so I developed a tool called FiberHMM, which is available on GitHub. And if you're doing any work with FiberSeq, I highly recommend using it. I'm still updating it. And basically, this allows us to then look not at these patterns of accessibility, but rather at the footprints of the chromatin-bound proteome on these long fibers. So that brings me to the outline of the rest of the talk. So first, I'm going to talk a little bit about what we can actually see when we visualize transcription in chromatin with fiber-seq and the kind of questions we were able to ask in the paper. And then also I'm going to go in a little bit more detail about one of the stories, which I think is probably more relevant to the seminar series, which is looking at how pausing influences the surrounding nucleosome landscape. Okay, so what does footprint in fiber-seq look like? So here we're going to be focusing on just sort of a random locus, which I think has nice example genes. So we're going to be looking at this gene RAU, and you can see that if we zoom out and look at these fiber-seq reads, we're covering like very large area. So each of these in this 12 kb region, a lot of these reads are spanning the entire region or are truncated on one side or the other. And so they're including not just RAU, but also these other genes upstream. But we can zoom in now on this actual promoter and see kind of the really exquisite detail that you can actually observe. So what we're capturing here are individual fiber-seq reads, which is each individual line. And I've marked on these fiber-seq reads the position of footprints of the chromatin-bound proteome, which I've also colored based off of their predicted identity. So each of these individual rectangles represents a so for example, the green rectangles represent nucleosomes, the pink ones represent pause pol 2, and the blue ones represent the pre-initiation complex. And then there's also some gray ones, which we didn't really identify for the purposes of this paper, such as those that are upstream, probably transcription factors. We identified these based both off of size and also off of orthogonal short read data, providing evidence such as ProSeq and CageSeq data. Speaking of this orthogonal data, if you just add up the distribution of these footprints, like the nucleosomes, the promoter proximal pause in purple, or the pre-initiation complexes in blue, they line up really nicely with orthogonal short read approaches. So the MNA-seq really tracks with nucleosomes, ProSeq really tracks beautifully with the pause, and CageSeq really tracks well with the pre-initiation complex. But we're able to see the states underlying these distributions. So for example, we can observe which fibers actually had an accessible promoter versus a inaccessible promoter. We can look at which reads have a promoter proximal pause, where the pause is, where the pre-initiation complex is. We can look at the positioning and the occupancy of the -1 nucleosome, which seems pretty variable at this particular gene. And we can look at the positioning of the +1 nucleosome, which seems to generally be positioned, but can vary from being positioned right at the TSS all the way out to 200 base pairs at this particular gene. And we can look at all sorts of other footprints, other nucleosomes, all at the same time. And the key here is that we're not just looking at these states in isolation, but rather we're looking at the interplay between these states on these single chromatin fibers, both at one locus and also at all the other loci that are captured within a given read. So we can ask questions, and these are some of the questions we asked in the paper, like, do we see co-occupancy of pausing and initiation? This is something that, unless you have single molecule data, you can't directly observe. And while it's been known that pausing generally inhibits initiation, here we can see that, at least at this locus, pausing seems to overlap pretty significantly with the PIC, and you can't really see them co-occupy. However, interestingly, at other genes, you do see them co-occupying on single fibers, suggesting that it really is just a steric effect that's blocking initiation. We can also look at the relationship between polymerase footprints at different genes. So as I mentioned before, we're capturing multiple genes within a single read. And so we can ask, do we see coupling of transcription initiation or pausing at pairs of genes, pairs of enhancers in genes, pairs of tRNA genes? And we do actually see that in a very distance-dependent manner. And again, you can read about that more in the paper if you're interested. But finally, I want to just mention what we're actually going to talk about a little bit more detail in this talk, which is how transcription actually influences the surrounding nucleosome landscape by observing both polymerases and nucleosomes on single reads. Okay, so let's look at pausing and nucleosomes. So this was really inspired by an observation that we made and that previous groups like the Edelman group had made with MNA-seq data, which is that when you look at genes with very low pausing, like a low pause index based off of ProSeq, you tend to see very ordered nucleosomes and very kind of accessible promoters. So here's 2 example genes, and I think it's fairly obvious that they look pretty distinct. In contrast, if you look at very highly paused genes, you see very disordered nucleosomes, a lot more variability in promoter occupancy, promoter accessibility rather, and a lot of Pol II footprints, which is what you'd hope to see. But you can see there's a lot of variation in +1 and downstream nucleosome positioning. We can look at this globally by basically turning FibreSeq into something similar to MNA-seq, where we're just looking at the pileup of nucleosome footprints. And you can see that at very highly paused genes in light green, we have very disrupted downstream nucleosome and upstream nucleosome landscape, whereas at more lowly paused genes in general, we have very ordered nucleosomes. And this isn't anything to do with the accessibility of the promoter, because we're actually controlling for that by only including reads where we had an accessible promoter. Okay, so what's going on here? Is this something to do with just this different promoter structure of highly paused genes? Or is this actually something that's directly caused by pausing? Well, we can start to get a hint of that because we can just filter our reads based off of whether they have a promoter proximal pause and test globally if we see a difference. And we have a hint that this might be the case because if you look back at this original example gene that I showed of a very minimally paused gene, when you do observe a pause Pol II at the few reads where you do, the reads actually look a lot like the reads that you observe at highly paused genes more frequently, where you have very disrupted nucleosomes. And it turns out this is sort of broadly the case. So if we just filter all of our reads based off of promoter proximal pause occupancy, controlling for the overrepresentation of highly paused genes, footprint is actually a little bit smaller as well on average. So it seems like there's quite a bit of disruption, specifically of the downstream nucleosome landscape. So you might say, okay, well, this just means that when you have a pause, nucleosomes are disrupted, and when you don't have a pause, they aren't. But in reality, we see a kind of more complex picture. So here's an example gene, and we're going to break down what states we're actually observing at this gene. So first, we have a population of reads where we just don't see any shift of downstream nucleosomes, and we don't observe a pause, but we can observe initiation. And then we have a population of reads where we do see shifted downstream nucleosomes, and those are a mix of reads where we observe a pause and reads where we don't observe a pause. So there's really 3 states that we're looking at, not 2. So how are they connected? Well, we can kind of come up with 2 models. The first is that this shift of the downstream nucleosomes is occurring prior to the pause, and then that leads to pausing being accommodated, and so that the shift basically is occurring prior to the pause and allowing for the pause. And this would be consistent with like a variation promoter structured highly paused genes. In contrast, what could be happening is that the shift is actually occurring with the pause, either driven by supercoiling or by chromatin remodelers, and that shift is actually somewhat stable after the pause, and that's where we're observing these reads where there's a shifted downstream nucleosome landscape, but no active pause. So what's the distinction that we can use to test these models? Well, in the first case, this is completely independent of the pause. And in the second case, the shift is going to be dependent on the pause. And so let's just globally change pausing. So we did this by treating with triptolide, which is an inhibitor that blocks the progression from the pick to the pause. And as you can see here from this meta profile of pause footprints, when you treat with triptolide, you lose this peak of pause footprints that you observe downstream of the TSS. And what we find when we treat with triptolide is that we lose this, we largely lose this shifted but no pause state that we are unclear whether it's caused by pausing or not. So if we're looking here, we're plotting the distribution of the distance to the +1 nucleosome, either untreated in color or triptolide treated in gray. And we're looking at different bins of pause index increasing as we go down. And what you can see is that the overall shift of the +1 nucleosome, when you don't observe a pause, increases as we increase pause index, but then when we treat with triptolide, it doesn't increase anymore, suggesting again that this is pause dependent. So that fits with the second model where pausing is driving this change. We can also test this model in a different way and add an additional validation, which is to test another assumption, which is basically that this is occurring after the promoter proximal pause has been released and entered into elongation. And if that's true, we'd expect to see more elongating Pol II footprints in this particular subset of reads. And that's exactly what we observe. So in the unshifted, unpaused state and the paused state, we don't observe very many elongating Pol II footprints. But when we specifically look at the state where there's no pause observed, but where the nucleosomes look like there's a pause happening, then you see a lot of elongating Pol II footprints. So overall, we have a model now that there's kind of more states of the promoter than I think had been previously appreciated. So we have sort of this closed state where, nucleosomes are blocking the TSS, and then that can be cleared and we have the ability to initiate, but there's not really space to pause. And then when pausing occurs, that drives this kind of general shift of downstream nucleosomes, and particularly the +1 nucleosome, which seems to be pretty stabilized. And then either at highly paused genes, this will be maintained by repeated pausing, or at less paused genes, we'll observe it less frequently because we just won't have as much pausing. And this shows a direct connection between pausing, nucleosome organization, and elongation. So in summary, we looked at a lot of other things in this paper. I would love for you to check it out if you want to especially read more about these coupling results. And I'd like to thank my various advisors on this and current projects. So Mike for being a great postdoc advisor so far, Sterling for advising me throughout my PhD, and Andrew for both developing FiberSeq and also working with me on this project. And I'd like to thank all three of their labs who have all been really terrific, but in particular, my co-authors, Stefan, Danilo, and Jane. And with that, I'm happy to take some questions.
Awesome. Thank you, Tony. That was really fantastic. I very much enjoyed your paper and then getting to hear you present the work as well. It's really beautiful. Um, I was wondering, have you tried with the FiberSeq specifically other ways of modulating pausing, like playing with NEL for CDK9? Or those like, are there like if you did like a PTFB inhibition, would you expect to see a lot more of the shifted nucleosomes?
I think that would be a really interesting thing to do next. That's something that was sort of outside of the scope of what we did, but yeah, I think one of the next steps here really would be to try to mess with pausing and see if we then see other effects there. Because yeah, I think that would be really interesting. Also to look at like chromatin remodelers as well and see if there's some stuff from Henikoff, Steve Henikoff recently looking at a BAF I know with pausing, and I'm a little suspicious of that maybe being involved.
Yeah, I mean, it's one of the like, is it polymerase itself, just the machine that it is that
Yeah,
sufficient? Or is it also recruiting remodelers to help?
yeah, exactly. Because it could just be supercoiling, right? You could just, by the action of transcribing like 25 base pairs, really kind of disrupt the sort of downstream sort of directional way. And I think that would also be a totally plausible explanation, but one that's also really hard to test because studying supercoiling, especially on like a single molecule way, is not easy.
Can you, with your data, when you see like a shifted nucleosome and the polymerase is off in the gene body already,
Yeah.
were you able to see how far the polymerase could be and still see the shifted state? Like to almost be able to infer how long is the shifted state maintained?
That's a really good question. I'll say that our confidence in calling elongating pole two footprints is not super high because we don't have as good evidence as we do for like a pause or a pick. And so I was hesitant to dig too deeply into, okay, if we have an elongating pole two at this position, where are things in that case? I think I would want to have a little bit more evidence that each individual elongating pole two footprint is actually an elongating pole two. But I do think that's a good question. I think it'd be very interesting to try to figure out a way to test how long the state is actually stable, because it seems fairly stable, and you'd imagine that it would require some energy to get things back to the original state. So yeah, I think that's another avenue that would be really interesting to follow up on.
Yeah. Um, all right. So then a few people have asked if pausing have effects on chromatin remodelers, which we've covered. And then
Yeah.
Just Brenner says, "Hi, Thomas. Thanks for your very nice talk and a really nice paper that I read thoroughly. Justice also uses FiberSeq and was particularly interested in the footprint sizes and was surprised to see such a wide range of the nucleosome footprint sizes from 100 base pairs to over 200 base pairs. And the question is about quality controls are used to make sure there's no under or over methylation in your samples. And if you'd seen anything like this.
Yeah, I mean, Andrew, when he was developing FiberSeq, did a huge amount of controls, just testing how responsive, like what longer treatments and higher treatments of HEA5 and stuff would do. And it really seemed very robust. Like it kind of gets to a certain point and then it doesn't really overmethylate. And as long as you do it to a reasonable degree, it doesn't really undermethylate too much. So I don't think the variation is really due too much to like any kind of experimental issues. I think it's probably more likely a combination of just the inherent kind of variation in the sequence context, right? Because depending on the amount of AT base pairs that you have, you'll have more or less accuracy in terms of the sizing. But also, I think there's a lot of variation in terms of how accessible, the nucleosome is going to be to HEA5. If it's very tightly wound, like what was being discussed with H4, the previous talk, then you might expect to not be able to methylate as easily on the ends. But, if it's more loose, if it's more unstable, then you might expect to see smaller footprints. And we do see a connection between smaller footprints at the nucleosome and this like +1 nucleosome that's kind of disrupted by pausing, which again suggests that there's probably some connection between the stability and the size.
Awesome. And then one other question is, like this you said was in Drosophila,
Yeah.
with a smaller genome. So how how feasible is this to do kind of genome-wide in mammalian cells with a much bigger genome?
Yeah, so it's definitely people. Andrew has done quite a bit of human fiber seq. I'm actually processing one of those data sets right now in the background on this computer. But yeah, it's definitely feasible. You obviously need to spend a bit more money on the sequencing. Looking at these kinds of longer range coupling is a little trickier, just because stuff is not quite as nearby, which is partially why I'm joined the Levine lab to really focus on Drosophila and really hone this while technology is improving. But I would highly recommend checking out the recent preprint from Andrew's lab on DAF-seq, which is a sort of different version of FiberSeq, which allows for single cell FiberSeq, which is kind of insane, and also targeted FiberSeq, which you can kind of tile in a way. And so I think the technology is definitely catching up to the point where it's going to be increasingly feasible to do that and to ask these like exact kind of questions in human cells. I guess the other difference is also that I think Drosophila just generally has more pausing and initiation happening. So it's a little bit easier to observe these with a lower overall level of coverage. So
Yeah, great. And I think Matteo has a question for you.
yeah.
Yes, fantastic talk. Just a curiosity about you joining the Levine lab. How feasible is it to do this in actual fly embryos? What's the input you require for the protocol?
Yeah, that's what I've been that's what I've been working on actually. Yeah, it's gone pretty well so far. Definitely have to prep a lot of cages, but we've gotten
Okay,
some data sets already.
so you still need, I guess, in the thousands of embryos range.
Yeah, so at the very low end, you can have like 50, 150 nanograms of DNA as input. But
Oh,
you're
okay.
going to be limited in terms of read length because you'll have to fragment the DNA a little bit more to get the molarity up higher. But with DAF-seq being PCRable, and then also with that, I think ideally you want like a microgram if you can get it, but if you can't, then there's ways to get around that, and it's pretty feasible.
Fantastic, thanks.
Awesome. Okay, well, I think we'll wrap up there. So thank you, Tommy, and our other speakers for a fantastic session. And thank you everyone for attending. We'll see you again in 2 weeks. Bye, everyone.